Good morning, everyone. I will tell you about our research regarding the role of the gene growth factor independent 1B, or GFI1B, in patients diagnosed with bleeding and platelet disorders. First, a small introduction on the formation of blood cells. Blood cells are produced in the bone marrow, and we have red blood cells that carry oxygen, and if you have a lack of red blood cells, you may suffer from fatigue. We have white blood cells. These cells are able to fight infections. And also we have cell fragments, which are called platelets. And platelets are essential to stop bleedings. And this is actually what our focus was on, because we were studying a family that had a platelet and bleeding disorder. And actually the patients could identify whether their offspring, their children had the disease immediately at birth, because if so, the patients or the newborn would have bruises all over the body. So to understand what caused this bleeding and platelet disorder, we wanted to discover the genetic defect, and also we wanted to understand how this gene would regulate platelet development. If you look at the platelet-producing cells in the megakaryocytes from the patients, we could see that the morphology of the cells was totally different in patients compared to healthy controls. You can see that the nuclei of the cell are not normally organized, and also the cell itself doesn't have this pink appearance, but is gray or pale. And this same phenotype could be seen in the platelets, because normally in a healthy control, the platelets are pink and have a normal size, but the platelets from the patient are gray and often enlarged. And this gray appearance is due to the lack of proteins that are very important in the function of platelets. And so the function of these platelets in patients was affected, but also the patients had a reduced number of platelets, and this combination led to the bleeding and platelet disorder that these patients had. And we found the cause of this defect in the patients, which was a defect in the gene growth factor independent 1B. So we have shown that GFI1B plays a crucial role in the development of platelet-producing cells and platelets, and only little is known still about the development of platelet-producing cells. We have shown that a defect in GFI1B can cause bleeding and platelet disorders. And during my talk this afternoon, I will discuss new genetic variants or defects in this gene. And these new genetic defects can help us understand how GFI1B functions at the molecular level. And in the future, because GFI1B is able to bind DNA and regulate the expression of other genes, and those genes are important for platelet production, we want to identify which genes are exactly regulated by GFI1B, which could be potentially therapeutic targets in this type of bleeding and platelet disorder, but also maybe in other bleeding and platelet disorders.